Hello, and welcome to our webinar on a high-productivity J.CHO expression system for the continuous manufacturing of antibodies, hosted by Evotec Biologics. Our speaker is Sherman Koo, Senior Principal Scientist, PNPD Cell Line Development at Evotec Biologics. Sherman received his PhD in molecular biology from the Scripps Research Institute after developing an iPSC-based disease model used for investigating the trinucleotide repeat expansion disease mechanism for Friedreich's ataxia. Afterwards, Sherman continued on to the Allen Institute for Brain Science, where he studied human brain development using engineered reporter iPSC lines, in vitro cortical neuronal differentiation, and various single-cell RNA-seq-based viral lineage tracing techniques. Since joining Evotec Biologics in 2016, Sherman has focused primarily on engineering J.CHO host cell lines with novel or improved functionality for biotherapeutic manufacturing. In his webinar, Sherman explains how Evotec Biologics is helping partners with candidate antibodies, Fc fusions, and bispecifics achieve the highest product quality with cost of goods manufactured of below $50 per gram by combining its brand new J.CHO High Expression System with its unique continuous manufacturing platform. This is followed by a Q&A session where Sherman answers questions from the audience. And now, ladies and gentlemen, enjoy Sherman's presentation.
I'll start off with an introduction to Evotec and our mission, then move on to a quick intro to our continuous biomanufacturing platform and what we think is a problem statement for what the industry needs. And finally, we'll hone in on our J.CHO High Expression System. So as we all know, foundational therapies often do not reach everyone, and these are specifically what I'm talking about are highly effective biologics molecules that are also unfortunately highly expensive for patients. On average, a biologic is around 22 times the cost of a small molecule drug. And as a metric for the skewed access to these therapeutics, roughly 80% of global sales of these monoclonal antibodies occur in the US, Canada, or Europe. And as of today, there are 7,000 underserved rare indications that could be addressed with biologics, but are considered non-viable from a business perspective. So I think this sums up a lot of the challenges that our industry faces in terms of access. Our mission then is to design and apply innovative technologies to dramatically expand global access to these highly effective biotherapeutics. And of course, one of the key levers we have to drive down the cost, to move the needle on cost, is our cell line and its productivity. And so to give you an idea of our company and our platform, specifically our J.CHO High Expression System, this is integrated with our continuous perfusion manufacturing. And as of today, our estimates in terms of reducing cost of manufacturing and cost of goods is a reduction by around 75% compared to a traditional fed batch process. And what that equates to a cost per unit mass is less than $50 per gram. This is always a moving target. We're always seeking to drive this down as well. In addition to product quality, we are also keenly aware that the product quality needs to be there in our product as well. Now, as we hone in on our J.CHO High Expression System throughout the rest of this webinar, there are generally two aspects that I think we'll focus on that make a host cell line highly effective one. One is productivity, and in our case, some of the highest examples of productivity that we've seen with our host cell lines is on the order of 4 grams per liter per day or higher in our perfusion platform. And the other important aspect is product quality, which I'll touch on in later slides. So one component of our J.CHO High Expression System, of course, is our expression vectors. And this is something that we developed probably beginning 6 or 7 years ago when we moved into a transposon-based expression system. So diagrammatically on the right, there's a scrubbed image of our proprietary vectors. But with some of the benefits that we see with using transposon-based expression vectors is that we see high levels of expression in both electroporation pools as well as single-cell clones. The way these transposases are integrated into our genome enables rapid cell line development. And this, of course, translates into very high specific productivities, in some cases greater than 50 picogram per cell per day for monoclonals as well as Fc fusion molecules. And then of note, one of the first BLA filed with this expression vector occurred in 2022. And we currently have 6 patents covering our proprietary vectors. And of course, the other component to a high expression system is the host cell line. And so what we've done is internally developed a glutamine synthetase knockout host cell line, originally obtained from a CHO K1 that we received from Larry Chasen in 2015. Over a year or two, what we've done was to take the CHO K1 cell line from Larry Chasen, wean them off serum, adapt them into our internal proprietary media. Then using a MAD7 gene-edited nuclease, we performed a biallelic knockout of the glutamine synthetase gene. From there, we single-cell cloned, selected a top candidate out of around 100 candidates, tested for IVV, mycoplasma, and bioburden. And from there, that became our established host cell line, or J.CHO high expression host. Since then, we've created several fully released MCBs, and in the last couple of years, we've completed 4 manufacturing runs. 2 of these were monoclonals, 1 bispecific, and 1 Fc fusion. As an example of the kinds of productivity that we're seeing, here's some data comparing using equivalent titer, which is a calculated metric, but comparing equivalent titer of a 14-day fed batch, comparing to our 15 as well as 25-day continuous perfusion manufacturing runs. And so as you can see, some of the highest productivities that we've seen in perfusion are somewhere between 6 to 10 times more productive compared to a 14-day fed batch. Nominally, these are 30 grams per liter up to 65 grams per liter. So that's quite a dramatic increase, and as we'll see in later slides. Part of this is from the continuous perfusion platform, but another significant portion is the increase in productivity of our host cell lines. Now, shown here, using a case study molecule MAB-A, what we see is that our J.CHO high expression host grows quite well. On the left is a plot of VCD, or viable cell density over time, and on the right is permeate productivity over time. What you can see is that our hosts grow quite well, in this case reaching our set VCD target of 90 million cells per mil. What's not shown is viability, but the viability also stays quite high in this case study molecule. Towards the end of production, the viability is still 90+%. And then, of course, the productivity, which is, again, one of the biggest levers that we have for driving down cost, you can see is quite high in terms of grams per liter per day, reaching a peak of almost 5 grams per liter per day. And in this case, because it's a 25-day run, where we begin harvest on day 7, that equates to 18 days of harvest, resulting in approximately 65 grams per liter equivalent of product. So you can see that is quite dramatic compared to a fed batch process. We've expressed a wide diversity of molecules using our J.CHO expression system, and we've given example titers here. From left to right, we have 3 different monoclonals. We've expressed several bispecific antibodies, as well as a handful of Fc fusion molecules. And obviously, there's a lot of range in the difference of titers, as we've all seen. Much of this is molecule dependent. But as you can see with bispecific molecule B, the 5th from the left, that one is a particularly difficult to express molecule. And even then, we're seeing over 1 gram per liter per day. Now, to dive in more into our cell line development toolbox, what I'll be talking about today are 2 of the 3 buckets here. One is a few-date knockout host cell line, and the second topic I'll be talking about is our inducible cell line. Sialic acid enhancement bucket, this is still early days, so we don't have any data to present yet, but hopefully this will be up and running soon. So jumping right into the FUDATE knockout. The idea here, as we've all seen, is that there's an increasing interest in producing molecules that are afucosylated. And the reasons behind this may be to increase antibody function, for example, with ADCC activity. Another big driver could be matching product quality if we're talking about biosimilars. And the approach we've taken here is to take our J.CHO high expression host, our glutamine synthetase knockout host, perform another biallelic knockout using MAD7. And what we've done is to knock out the fucosyl transferase 8, or FUDATE gene. This, of course, encodes an alpha-1,6 fucosyl transferase, which catalyzes the transfer of fucose from GDP fucose to its target sugar moiety. There's a diagram here that I've taken from a paper on the right, probably oversimplified, but this is a fairly well-characterized gene in the literature. So I'll skip over probably a year and a half of characterization data, PCR sequencing. In the next few slides, I will show some highlights from a comparability study. What we want to ensure is that these genetic knockouts perform very similarly, yet has its intended effect from the FUDATE knockout itself. So using case study molecule MAB-B, what I'm showing here are growth performance metrics, VCD as well as viability, comparing our J.CHO host, our parent GS knockout host shown in green, and our FUDATE knockouts, but really a FUDATE GS double knockout in orange. And the key takeaway here that I'd like to convey is that the growth performance is quite similar in terms of VCD as well as viability. And for the sharp-eyed folks in the audience, you'll note that the VCD does decline as well as viability over time, different from the data that we've shown several slides ago. This we know to be very molecule-specific. In most cases, our bench scale reactors, our viabilities don't decrease beyond 90% by the end of production. And usually we do hold our target cell densities very consistently across the production run. Now, looking at productivity, using productivity, permeate titer and specific productivity, and various calculated metrics, we can also show that the productivity of our FUDATE knockout is quite similar to our J.CHO parent. Again, using the same colors, we see that productivity peaks around 1.5, maybe 1.75 gram per liter per day. And if anything, this data seems to suggest that our FUDATE knockout may be slightly higher in terms of specific productivity compared to its parent, if we're looking at the plot on the lower right. However, the difference here isn't that significant, and with other molecules, we see the specific productivity difference to be even smaller. Some of the data shown here is unique to this MAB-B. But nonetheless, productivity performance looks to be quite similar. And finally, perhaps the thing that we're interested the most in terms of a FUDATE knockout is seeing if we have an effect in terms of the glycan profiles. And using HILIC, we can look at the degree of afucosylation, silation, the amount of beta-galactose, as well as total high mannose. In the top left plot, this is perhaps the thing we're most interested in, looking at the percent of afucosylation. And here we see a very night and day, very dramatic difference between our FUDATE knockout and our J.CHO parents, again in orange and green. And so note the y-axis scale here with a FUDATE knockout showing close to 100% afucosylated versus the J.CHO parents. The data would suggest that the degree of afucosylation is not 100%, but we believe this is a consequence of the HILIC assay and chromatographic peak integration. In the next slide, we'll have other data to support that the afucosylation is, in fact, quite effective. Other glycan profiles, such as silation, total high mannose, on the right column, seem quite similar to its parents. Note that in the top right plot with silation, the y-axis scale is quite small. So while the data appears to be quite different, the difference between 0.5 and 2% acetylation is not huge. Now, interestingly, what we see in beta-galactose on the lower left panel is that there is a significant difference due to the FUT8 knockout. And what we believe here is that this is perhaps a cascading effect from the FUT8 knockout itself. And as a proof of principle, what we've done here is to express rituximab using our FUT8 knockout host cell line, purified the product, and characterized them both by SPR as well as a cell-based functional assay. In both cases, what we see is afucosylated rituximab shows much higher activity compared to the version of rituximab using the J.CHO parent. In the right plot, you can see a difference between the blue and green traces, and by SPR in the table in the lower left, you can show the binding kinetics of afucosylated versus the native version. So with that data, do you feel like we have enough data to show that the FUT8 knockout is quite effective? And so this cell line is now one of the J.CHO High Expression cell line hosts that we offer, and is currently available for use. Now, moving on to our inducible cell line host. So the idea here, driving the development of this inducible host cell line, is that the expression of exogenous protein or antibodies increases metabolic burden. And as an effect, cells with lower protein expression often will outcompete or outgrow cells that have high protein expression. This is a phenomenon that we and others have published. This is a pretty widely seen effect. And so we decided to see if we can develop a host cell line where we can control, or in other times limit, the expression of our exogenous protein. And the idea is that with constitutive expression, shown in these cartoon figures in the center, is that over the course of electroporation and recovery from drug selection, what survives that drug selection often is inherently adapted to survival or adapted to growth, given an extrametabolic load. And so the idea is that with constitutive expression, whatever survives drug selection is inherently adapted to survival and optimized for growth, given the extrametabolic load. And perhaps there are subpopulations in your cell population where there's so much protein expression, so much extrametabolic load that they do not survive expression. And those we've indicated in the dotted red line. And so those are cells that don't survive, and we lose them, and we're not able to clone those. Now, with an inducible expression, what we aim to do is to limit or completely reduce expression of your exogenous molecule, antibody, bispecific, what have you. This removes the metabolic burden from cells during a relatively stressful time of drug selection. And so whatever high producers there may have been, in this situation, we believe that they will survive selection and become accessible for us, the single cell clone. And so the platform that we've used is based on the tetracycline responsive system. This requires there to be a presence of a TET-R responsive protein that binds to a TET-O operator in our expression vectors. And in the presence of DOX, this undergoes a conformational change and allows expression of your protein of interest. This is very well established in the literature, so I won't go into many more details beyond that. And again, I'll skip over a lot of comparability and characterization data and show some highlights here. And what we really want to show is that our inducible host cell lines, they grow and perform quite similar to the parent J.CHO high expression host. What's shown here are cell growth plots. On the top is VCD, and on the bottom is viability. And so again, looking at the same colors, comparing between the parent in green and the inducible in orange, we see that cell growth is quite similar. And in this case, we're holding the target VCD quite well. Viability is also pretty similar, comparing the inducible versus the parent. And if you look carefully, looking at the square points versus the circle, those are the differences between a no DOX and a DOX condition. And so we've also shown, using this data, that doxycycline does not affect cell growth. And then looking at productivity, this is perhaps where we see the biggest difference. In these case study data, this is based on an Fc fusion molecule A. What we're seeing on the top plot in productivity is that there's very little productivity, both in the parent or the inducible. But when we dose at day 7, where the red line is, when we dose with doxycycline, that's when we see a dramatic increase in productivity. And that's shown in the orange circle points. We can see a peak of around 1.7 or so gram per liter per day. And I'll note that this is a particularly toxic molecule to express, very difficult to express. But we've shown that with this particular inducible cell line, that we can ameliorate a lot of the effects from the toxicity of this molecule. And finally, looking at product quality, again, at glycan profiles using HILIC, we can see that these profiles are generally quite similar between the J.CHO parent as well as the inducible. Looking at afucosylated, percent afucosylated on the top left, sialation on the top right, and then beta-galactose on the bottom left. These are all pretty indistinguishable comparing the parent and the inducible. And then interestingly, on the lower right panel, looking at total high mannose, we do see an increase with the inducible cells with DOX induced. This we believe to be a consequence of the molecule being toxic. We do see similar effects in viability, and I think we've all seen correlations between viability or unhealthy cultures and high mannose. We believe this to be very molecule specific, and in other cases, we see less of a dramatic effect here. And so to wrap up, that is our inducible cell line platform. This is also completely characterized. We have RCBs of this cell line. They are available for client use as of today. And like I mentioned earlier, the sialic acid enhancement, or the more glycoengineered host, this particular bucket, this is early days, but this is something we're actively working on and something that we hope to make available to our clients quite soon. But the longer term vision here is that given a particular product quality profile at an innovator, and we can choose the correct engineered host or to tune or maybe match the product quality of a particular innovator. And so to wrap up, what we've seen here today is that the Fudate knockout host that's now available performs quite well and quite similar in bioreactors to our J.CHO parent. Product quality is reasonably similar. And the same with the inducible cell line. Growth and product quality is quite similar. And the added benefit here is that we can control protein expression, especially if there's a cytotoxic molecule that we're dealing with. So, of course, that's only the host cell line and the transposon portion of our J.CHO High Expression System. One other component that I haven't touched on today is our internal proprietary media, and altogether these three integrate into a highly performance platform that's available to all our clients. So with that, I'll stop here. These are the folks that were involved in this work. The names in italics are major contributors to this work. Many thanks to them. None of this would have been possible. Thanks so much for your attention.
Thank you very much, Sherman, for your great presentation. And now, ladies and gentlemen, Sherman answers questions from the audience.
How many VVD are we running during harvest? And do we have a proprietary perfusion media? Short answer is yes, we do have a proprietary perfusion media. And for VVD, we run at 2 VVD. We have a ramping period of, I think, 2 days where we increase from 1 to 2 VVD. But once harvest begins, 2 VVD through the end of production. During CLD, are we selecting clones based on specific productivity and simulated perfusion? Or does fed batch translate to perfusion systems at scale? Yeah, great question. We select based on using a fed batch production currently. And this isn't a perfect model for our perfusion platform. And we know that growth profiles of viability, the peak DCDs, aren't anywhere close to what we see in perfusion. But having said that, we've seen that the relative performance of clones that we choose in fed batch does translate to relative performance in perfusion. So in that way, using fed batch is a way for us to screen clones. We are currently looking at mock perfusion or simulated perfusion, as you're calling it. This, I think, has some other benefits that we're investigating. I think, of course, it's a much better growth model. In some exceptions, we've seen that we actually select better clones using mock perfusion. However, the way the experiment is set up, at least for us, it's a little bit less amenable to scaling with liquid handling automation. I mean, that's not actually something I didn't touch on at all. But a lot of this work, we heavily rely on automation, which allows us the throughput that we have. In the case of MAD-B, where a significant drop in viability was observed with cell culture duration, how do we anticipate it would perform in the inducible host? The way that the viability dipped with MAD-B, it occurred well into harvest. If I remember right, it was around day 9 or 10 when it started losing viability. So my sense is that using an inducible cell line may not have helped in that case. That particular molecule, I think we consistently saw trouble with growth. With an inducible cell line, I think the benefits we get there is that we can ramp up cell growth quite well without this extra metabolic burden. But once they hit VCD and once we induce with doxycyclines, I would suspect that we would still have a similar viability issue with that particular molecule. So one of the questions that I frequently get is around IP. With the MAD7, the license that we have gives us a royalty-free ability to make any kind of gene edits. The timing at which we obtained the MAD7 protein sequence was quite lucky. And at the time, this was maybe 6 years ago, their license was pretty open. Other aspects include the few-date knockout. We've done a pretty thorough investigation on that. We do have freedom to operate there, and the same with the tetracycline inducible system. And that's one of the things that we've been pretty intentional about. The goal of our company is to drive down costs of therapeutics. Then we're doing a lot of things that we can to avoid things like patent or royalty fees. Are we able to monitor glycosylation and perfusate in real time to stop harvest? We don't. It's near, I would say it's not real time. We sample at the day of culture. Typically how we work is that we collect all the day of culture samples, save them until the end of production, and then submit them all at once. And so obviously that's very not real time. But if we were to analyze glycosylation the day of, it would still be about a 2-3 day turnaround. So at the moment, I would say, unfortunately, we don't have that ability to control when we stop harvest. But that is our cell lines specifically designed for perfusion. How do they perform in fed batch? We haven't specifically engineered anything or done any selection for hosts that perform in perfusion. Our platform also is perfusion. In our early days, we have run certain cell lines in fed batch. And while those perform reasonably well, one of our cell lines that we sometimes will use is the HD-Bio3 cell line from Horizon. If I remember right, some of our earlier work comparing these two in fed batch was pretty comparable. But as a company, as a platform, we are heavily invested in perfusion. And we do believe that that's the way to increase productivity. Do we see any variability in terms of charge variance with continuous manufacturing? That's a great question. We do see some fluctuation. In some cases, this is molecule dependent. I would say generally, especially during the time frame of harvest, when VCD is very steady, viability is very steady, we see pretty stable charge variance, I would say. For the 25-day expression, do we start harvesting starting on day 7, and then do you add more media to replenish the culture? Yes, we do start at day 7 or 8. And I alluded to this with one of the earlier questions. We do perfuse in 2 VVD of fresh perfusion media as we're harvesting. Can the continuous cell culture go longer than 25 days? I think it can. We haven't done much work on that. Usually with the client projects that we have, we get enough material from a 15-day perfusion, and in other cases, a 25-day perfusion is more than sufficient. Have we done any cost of goods analysis with your cell line and the range of MABs that we've developed? We have. I don't have the data off the top of my head, but we do have an entire cost modeling team that tracks this pretty closely and makes sure that we're on track and still trending downward in terms of cost. Was all the data shown today using our proprietary media? Yes, that was all proprietary perfusion media. For growth, I didn't mention this at all, but for passaging, for growth, we use a commercially available CD-Opti-CHO media.
This is the end of our event. Thank you very much for your participation. If you would like to get in touch with our speaker, please email us at info@evotec.com.